The ratio of adenine to thymine in DNA is 1:1. A always pairs with T. T always pairs with A. And those were from Chargaff's rules. He realized that those numbers were always the same. Likewise, guanine is always equal to cytosine. Cytosine is always equal to guanine, because G always pairs with C. C always pairs with G. And so in all DNA, regardless of species, because here we have beef thymus, liver, beef sperm, human thymus, human liver, red blood cells, herring sperm, wheat germ, yeast, it's always a 1:1 ratio. A always pairs with T. T always pairs with A. C always pairs with G. G always pairs with C. And Watson and Crick and Rosalind Franklin worked out the structure of DNA and realized it was in a double helix. And A's and T's bind, and if you look at it, notice the hydrogen bonds. A's and T's have 2 hydrogen bonds between them. C's and G's have 3 hydrogen bonds in between them. And those hydrogen bonds keep the double helix together, but it's also relatively easy to unzip them, to copy them. And so once they realized the structure of DNA, it was immediately intuitively obvious how the DNA should be copied. All right, the structure of DNA, again, A always pairs T, and there'll be 2 hydrogen bonds. There and there. In C and G, C always pairs G, G always pairs C, 3 hydrogen bonds. They actually hold on to each other a lot tighter. So in the origination spot in bacteria, for example, when you're going to open up the DNA to copy it, it's usually very rich in A's and T's because that's fewer hydrogen bonds. It's an area where it's easier to unzip the DNA. And the DNA is always complementary because A always pairs T, T always pairs A, C always pairs G, G always pairs C. If you know the sequence of one strand, you can figure out the other strand. If I gave you A, you know the other strand has to be T, 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 T. If I gave you GA, you know the other strand has to be CT, CT. Because if there's a G, the other one has to be a C. If there's an A, the other one has to be a T. If there's a C, the other one has to be a G. If there's an A, the other one has to be a T. If there's a T, the other one has to be an A. And so they're complementary. So if you know one strand, you can deduce the other strand. Human DNA, this is a molecule that can be extended almost 10 centimeters in length. But it's contained in a nucleus with a diameter of 10 to the negative 6 meters, a micrometer. The DNA is looped around small aggregates of positively charged histone proteins. Histones will become really important in epigenetics because a histone protein has a tag. It's a lysine, a moiety from an amino acid. And if you methylate it, that nitrogen will become positively charged. It'll bind the negatively charged DNA, and then that is not unwrappable. It's not open-upable, and so that DNA is turned off. Whereas you can remove the methyl groups from the lysine and put an acetyl group there. And now there's no charge, and that is an open piece of DNA, and it can be opened up and read. And so epigenetics is a science of what genes are turned on and what genes are turned off in eukaryotes. And that's done at the histone. So that's actually really important in genetics and epigenetics. Further condensation of the DNA molecule is achieved by wrapping the structure into a tight coil called a solenoid. So here we see the solenoid structure. DNA wraps twice around a histone. It's about every 200 base pairs you find a histone. And so this is kind of like bubble wrapping. The DNA is the most precious thing in the cell, so it wraps it very carefully and exquisitely to make sure it doesn't get damaged or hurt because it's irreplaceable to the cell. A sample of DNA contains 26% thymine. What is the amount of adenine in this sample? It'll also be 26% because it's 1:1. 35% of the bases in a certain DNA molecule are found to be thymine. Which ones will be guanine? Well, if 35% are T, then 35% will be A. So 70% will be A and T, leaving 30% for C and G. So we divide that by 2, 15%. 15% of the bases will be guanine, 15% of the bases will be cytosine, because your overall total's got to be 100%, and A will always equal T, C will always equal G. Hereditariness. This is a process by which the physical and mental characteristics of parents are transferred to their offspring. Mendel is the father of modern genetics, and he actually worked out hereditariness in genetics before genes or chromosomes were even discovered. And so in his writings, he just called them factors. They were informational factors of how because he wasn't totally sure exactly what molecule was carrying the information, but he saw that information was transferring from parents to babies with his pea plants. So the genetic information needed for transmitting a species' characteristics is coded in the DNA. The genome is the sum of all hereditary material in the cell. All of your DNA combined is the genome. Bacteria have one circular chromosome. Eukaryotes have many linear chromosomes, and those numbers of chromosomes can vary between 2 to 1,000, but they're linear, not circular. A gene is a segment of DNA that produces a functional product. So a gene will produce a functional piece of RNA. That RNA may be messenger RNA, which will land on a ribosome to make a protein, or that RNA may be an actionable product in and of itself. A gene can produce either a protein or an RNA. They're both necessary and useful by the body. So in general, a gene is considered a segment of DNA which produces a useful product that does biochemistry of some sort. Replication. This is when DNA doubles. You have a strand of DNA and you copy it, now you have 2 strands of DNA. And what you see in the picture is semi-conservative replication. Each new strand of DNA is half new DNA and half old DNA. So it gets one old blue strand, but also it gets one new red strand, each one. So the DNA content of a cell doubles before the cell divides, and that occurs in the S phase of the cell cycle. And then you go through the G2 phase, in which they proofread the cell proofreads the DNA and make sure it was copied correctly, and then the cell will go into mitosis and double. Again, reproductive cells undergo meiosis. So replication in reproductive cells, you actually have 2 cell divisions, and you end up with 4 cells that only have half the correct number of chromosomal information, so that an egg and a sperm can fuse together to form a new person. So the number of chromosomes varies within species. Humans have 23 pairs, or 46 chromosomes. You get 23 from your mom, and you get 23 from your dad. So humans have 23 pairs of chromosomes. You get a number 1 from both your mom and dad, a number 2 from both your mom and dad, a number 3 from both your mom and dad, all the way through all 23 pairs. You get 1 from mom and 1 from dad. How many chromosomes are in human egg and sperm? Well, they only have half the number of chromosomes, so they only have 23 chromosomes. They're not in pairs. They don't have 46 like a full cell. And that way an egg and a sperm can come together, and now you do have 23 pairs. You get 1 of each from the egg and 1 from the sperm. Cancer cells and DNA replication. Cancer cells are fast growing, and basically it's a growth disorder. A cell is growing too much, too fast, and it's not listening to outside cues telling it that not to grow. And so we use cancer chemotherapies to fight cancer. And a lot of these drugs interfere with DNA replication because cancer cells typically divide the fastest of all cells, and so medications aimed at very quickly dividing cells are effective at chemotherapy. Topotecan is a drug that blocks the unwinding of DNA, which is necessary for replication. Some flat molecules like cisplatin and adriamycin slip in between the DNA bases and disrupt replication. Gentamicin and Gemzar are unnatural nucleosides that cause an abortive DNA replication, and the DNA is not copied correctly and you don't get another cell. RNA. One of the main functions of DNA is to direct the transcription, which is the synthesis of RNA from DNA. And RNA consists of single polymeric strands of nucleotides. It is not a double helix like DNA, it's just a single strand. And we have ribose instead of deoxyribose. And again, we use uracil in RNA instead of thymine. Some types of RNA have a significant number of modified bases in addition to the common 4. Function of RNA. We process genetic information into protein. So messenger RNA is just like a burnt CD. It's a copy of a gene that's allowed to leave the nucleus and land on a ribosome and be made into a protein. And it's a disposable copy, because if it gets destroyed, you can just make another copy in the nucleus and send it back out. The DNA is super precious. The DNA is never allowed to leave the nucleus. And therefore, it makes these disposable copies that are allowed to leave the nucleus to do stuff. Ribosomal RNA, which is produced in the super dark staining area of the nucleus that we call the nucleolus, makes up the majority of a ribosome. The ribosome just has a thin protein shell. Think of an M&M. The ribosomal RNA is like the chocolate of an M&M. It's the core of the M&M. And the protein of a ribosome is like the candy coating. It's just a superficial coating. So the guts of a ribosome are actually ribosomal RNA. And that's made in the nucleolus of the nucleus and then exported out to the cell to produce ribosomes. And transfer RNA. This is the tRNA. It shuttles in the correct amino acid, and then the anticodon of the tRNA matches a codon in the messenger RNA to make sure the right amino acid goes in the right space while you're making your protein. And there's at least one transfer RNA for each of the 20 amino acids, which your DNA codes for. And if you get into a more advanced biology class, you'll see that there are a couple more amino acids that your DNA actually has nuanced codes for, like selenocysteine is typically called the 21st amino acid. Your body will first put a serine in, and then later modify the serine into a selenocysteine. So there's lots more to learn there with DNA and proteins.